Hello, my name is Darcy Taniguchi, and I'm an assistant professor at a university in California called California State University San Marcos, and it's located in Southern California. And in the year 2009, I was lucky to be a part of a voyage on the New Horizon, which is a ship. And we went out, me and a group of scientists and media professionals went on this expedition to look at plastic in the Pacific Ocean. In particular, we were interested in seeing how it's laid out as we go from Southern California out into the Pacific and then back towards land. And the particular organisms that I was interested in studying in the ocean are organisms that are made of only one cell. They're called plankton. They're the drifters of the sea. And I wanted to see where they're found in the ocean and how they may be affected by plastic in the ocean. How do you describe a 24-hour day for you while working on the New Horizon in the Gyre? So my day actually starts in the evening. I would wake up for dinner, would be my first meal of the day, and I'd go to the galley, make sure I had a full stomach of food to prevent any sort of seasickness, and then I would go set up the lab to get ready for my sampling for that evening. After that, I'd grab my pen and my data sheet and go to a whole suite of computers. I decide where exactly I want all of the bottles on the CTD rosette to close. And after I close them all the way up, then that rosette would come back on shore on the deck of the ship, and I would go out with all my many bottles and sample water that was collected from a whole variety of depths in the Pacific Ocean. Once I had my bottles full of water, I'd take them into the lab and then process them, filter it down, and put it on a glass microscope slide that I'd look at back on land. After I did all that processing, then it would be about time for breakfast. So I'd go, and if I was lucky, there would be fresh baked goods like muffins already ready before breakfast, and I'd steal some and get a little bit of sunlight, because remember, this is all happening during the nighttime. And then I'd have a snack, enjoy some sunshine, maybe have breakfast, and then get together my data sheets, my notes before going to bed. That is a CTD rosette, or a conductivity temperature depth rosette. So at the bottom, actually, in the middle of that bottom ring, you can see there's this black part and this silver piping. That's where the sensors are to measure the salinity, the temperature, and the pressure as a measure of depth in the ocean. And then the big yellow part is just a frame for each of those bottles, which are the long gray cylinders. And what's really neat about this contraption here is that all of those bottles can close at whatever depth we want in the ocean, and it's a way for scientists to sample particular depths of water and then bring it up on deck and see what's there. I would go set up the lab to get ready for my sampling. For that entail labeling little bottles or little vials where I'd be putting all of my water samples, getting my forceps, my tweezers ready, and places where I'd be putting the water and processing it. After that, I'd grab my pen and my data sheet and go to a whole suite of computers, a lot of different computer monitors set up. And that's where I would talk to the crew members who were vital to this expedition, and they would direct the movement of the CTD into the water. They'd help move it into the ocean, and then I would sit at the computer and look at how the different environmental measurements, particularly the depth, the temperature, and the amount of chlorophyll in the water, would change with depth. And based on that information, I decide where exactly I want all of the bottles on the CTD rosette to close. And after I close them all the way up, then that rosette would come back on shore on the deck of the ship, and I would go out with all my many bottles and sample water that was collected from a whole variety of depths in the Pacific Ocean. Once I had my bottles full of water, I'd take them into the lab and then process them in lots of different ways. Sometimes I just want to look at the chlorophyll. Sometimes I want to look at the bacteria that are able to photosynthesize. I want to look at all types of different single-celled plankton in the water, in which case I'd have to put different stains on them, filter it down, and put it on a glass microscope slide that I look at back on land. And the overarching reason why I sample at night versus during the day is because the organisms that I study have that chlorophyll in them. It's a pigment that they use to capture light. So it's really sensitive to light. And I don't want it destroyed by bringing them up out of the water into the bright sunlight during the day that would damage that chemical in them. So that's why I have to work at night. There were several challenges that we encountered, one of which that really has stuck out in my mind when I think about this question is just the of needing to sample all the time. So we would put down a rosette like every day and have to sample all of those bottles every day and go through the process of picking the water, bringing it into the lab and processing it so we can study those single-celled organisms. And that amount of really detailed technical work on a very regular basis, and admittedly with little sleep, is a very challenging aspect of being at sea. Another challenge in general for studying the organisms that I do, they're all microscopic, and so we can't see them with the naked eye. We need tools to assist with that. And because they're so small, they're often very hard to identify as well. And having technology to help in that realm would be very beneficial.